What's up, Science Fam? Last time we talked about some of the cellular structures, and this time we're just going to continue describing the structures inside the cells. My name is Sir Ramon, and this is Science with Sir Mon. Let's get right into it. What if it's time for the cell to split, just like you and your significant other? So what helps the cell divide? That would be the centrosome. So this is only found in animal cells. So it makes fibers for splitting chromosomes and making cilia and flagella. So your centrosome is actually made up of two centrioles. So they kind of look like churros. So you can see them in this image here. And these churros are themselves made out of triplets of microtubules. Now, this centriole is also known as a microtubule organizing center, an MTOC or MTOC. So they organize new microtubules in order to use them to make fibers, such as, again, for cell division when you split the chromosomes, and to make the cilia and the flagella, which are other types of structures. So, speak of the devil, what about movement? What helps the cell move around? That's precisely your cilia and your flagella. So your cilia are usually used to just move the stuff outside the cell, and they're smaller than the flagella. The flagella, on the other hand, are used to propel the cell itself. So flagella for locomotion, cilia for moving stuff that's on the surface of the cell or outside of the cell. So just to drive the point home, this is what they look like in action. So you can see the cilia just moving stuff outside the surface of the cell. Whereas the flagella, it kind of rotates like a propeller of a boat so that the cell moves forward or wherever it's supposed to go. Alright, next. Where does the cell get its energy? What powers these processes? So the energy of the cell comes from these structures. For eukaryotes, all of the eukaryotes have mitochondria, or mitochondrion for singular. It's the powerhouse of the cell. So it makes energy in the form of ATP or adenosine triphosphate. We'll get into detail on that later in the quarter. The chloroplast, on the other hand, is for photosynthesis. So mostly it's the plants that have them, but in the future, you might find that there are some protozoans or microscopic animals that actually also have chloroplasts. There's also the little leaf sheep thing. So I think leaf sheep is its name. So it's a sea slug that also has chloroplasts in its cells. So the chloroplast allows the cell to make sugar through photosynthesis. It makes food through photosynthesis. Now, these two structures have things in common between them. First, they have both an inner and an outer membrane. So they have a double membrane thing. So also present in the chloroplast. And another thing is they both have their own DNA, which is separate from the nucleus. So these two things are very similar to another organelle, your nucleus. So again, these organelles, the mitochondria and the chloroplast, they have some aspects that are similar to a nucleus. They have their own DNA and they have double membranes. This points to something. This is a clue to a certain theory that you guys will be searching about in your brainstorm discussion. So next, how does the cell process materials? How does it make different kinds of molecules? So that is done through the endomembrane system. It produces and transports biomolecules. So what first makes protein in your endomembrane system? Protein is very important for enzymes and structures. So what makes your protein is your ribosomes. So therefore protein synthesis, and they are present in all types of cells. So they synthesize or make proteins out of amino acids. Again, amino acids are the building blocks of proteins. Your ribosomes can be free floating in the cytoplasm, just there, around, floating around, or embedded in another organelle, which we'll see later on. So these ribosomes are actually made up of RNA subunits, meaning they're made up of ribonucleic acids. So again, they are assembled in the nucleolus. So very important, tiny things. So where does the processing begin? Don't we need instructions for this processing? And those instructions come from the DNA, so we need the help of the nuclear envelope. So the nuclear envelope is part of the endomembrane system because it transports RNA, ribosomes, and proteins into and out of the nucleus. So it keeps the DNA very safe from the possibility of being damaged by radiation and other things that can damage DNA. For bacteria, for your prokaryotes, they don't need this nuclear envelope because they want that damage to happen because it allows them to evolve faster. It's not as dangerous for bacteria. So next, where are lipids made and where are proteins processed further? That is in the endoplasmic reticulum, which is actually continuous or connected to the nuclear envelope. So again, if you look at the image here, it's actually connected to the nuclear envelope. So connected to it, again, the endoplasmic reticulum, it's the rough and the smooth. So there's two types, rough and smooth. The rough one is rough because it's embedded with ribosomes. Since ribosomes are for making proteins, the function of your rough ER is to fold and transport proteins. Your proteins are like very, very long chains, and they can't do their specific function unless you fold them into the proper shape. So your rough ER helps to fold them into the proper shape and then transport them where they need to go. Your smooth ER, on the other hand, transports lipids and detoxifies certain types of molecules such as ethanol as well. So it transports lipids, so things that you can't dissolve in water, and then it detoxifies things like ethanol. So it doesn't have any ribosomes, and that's why it's smooth. So how do these things get where they need to go, especially if they need to go outside of the cell? We have the Golgi apparatus. It sorts, modifies, and packages these different biomolecules. So it has some parts. We have the cis phase on this part and the trans phase on the other end. So your different proteins and lipids go through the cis phase and then all through these different pancake-looking things, which are basically cisternae. So it goes through different cisternae, which each modify these vesicles in order to make sure it gets to the appropriate place where it needs to go. So it uses, the Golgi apparatus uses vesicles just like Lalamove. So when it goes through the different cisternae, there are markers and addresses that are put onto the vesicle so that the vesicle gets transported where it needs to go. What else is part of the endomembrane system that maybe we've already discussed? So these are some of the things we've discussed that are also part of this endomembrane system. The plasma membrane is the final border for stuff that's going in or out of the cell. So it's like a border patrol. We'll talk more about this border patrol in later in the quarter. Vacuoles and vesicles, on the other hand, are like warehouses and messengers, respectively. Your vacuoles store things for long term, while your vesicles, again, as we mentioned earlier, they're like courier services. They're like Lalamove or Grab. Lysosomes are recycling centers. So if you don't need something, you can break it down into usable parts and recycle these reusable parts. So the cell is basically like a full-blown industrial factory. And it's no wonder why viruses want to take them over, because they can survive on their own, unlike viruses. But even if they can stand alone and survive on their own, cells work better when they work together. And maybe we can learn a bit from cells. So even though we're far apart and we can sort of kind of survive as standalone units, as people, as individuals, we can achieve a lot more when we work together. If you have any questions, hit me up through Messenger or the other ways we can contact each other. I'll see you guys in the next topic.